chloroplast genome has been constructed from scratch. So I think this is our 1995 for synthesis, meaning in 5 years I would be surprised if the construction of bacterial genomes and eukaryotic chromosomes like yeast isn't a routine process. It's hard to appreciate surfing along an exponential, even though we're all familiar with it from computing. When you see it playing out in biology, it's somewhat astonishing. So, for example, to come back to this paper where we engineered a 12,000 base pair fragment of DNA, here's the instructions for how we did it. You could find this freely available online under Creative Commons license. So you can read our instructions. To build the first section, we cloned parts 5, 6, 7, 8, 12, 13, 14, 15, 15, 16, 18, 20, 22, 24 into PSP 104. That's the best technician in my lab working for 3.5 months to execute that one sentence. We cloned part 11 into PSP 2K3. We cloned each part with its part specifically for acting restriction sites. Blah, blah, blah. You could read the rest. So this paragraph is 3.5 years of work to make a 12,000 base pair fragment of DNA. This is a project that ran from 2003 to 2004. Today, this paragraph gets replaced with the sentence, where you ship the sequence information over the web and somebody builds it for you. If you type in, you start looking for that online, you'll find lots of companies. Okay, I'll move quickly. Most genetic engineering takes place at this level, at the level of machine language. All right, so TAA, TA, CGA, CTC, ACT, ATA, GGG, AGA. 23 base pair sequence of DNA, which you'll remember is the consensus promoter for the T7 RNA polymerase, an enzyme that turns on transcription of. Now, I happen to remember this one sequence because it was my computer password for a while. But the problem is there are a lot of sequences like this to memorize, and if one were to program DNA at this level, it would be like only programming in machine language, I guess, if I'm using the right analogy.